Okay, let's go over chapter 13. We're talking about the muscular system, three types of muscle tissue. That's going to be cardiac muscle, your heart, and striated muscle. Sorry, those are both true. That's true, but cardiac muscle and skeletal muscle, which both of which are striated. That just means they have stripes. It's because all the fibers line up in the same way, and so things that minor tiny structures that you can't see if the fibers are all which way, they're easy to see. I'm talking about lines of actin and myosin, and we'll get into that. But anyway, the first two, the cardiac muscle, the skeletal muscle, those are striated. The third type is smooth muscle. That's involuntary and you can't see the striations. It still has the same fibers, but they're not lined up real pretty, so they're easy to see. Um, anyway, but smooth muscle is the muscle lining your blood vessels, lining your digestive system. That's what controls the diameter of your blood vessels or what moves food along through the digestive system. Um, that's involuntary. We cannot control it. That is also true of the heart. You can't control the heart. So cardiac muscle, involuntary. Um, anyway, describe the general structure of a skeletal muscle. This is where we're going to talk about myofibrils and stuff like that. And we'll talk about how muscles are named. Um, a lot of them are named based on where they're attached to bones, starting at the origin and going to the insertion. An example of that would be your sternocleidomastoid, named for the sternum where it attaches here, and the mastoid process of the skull where it attaches here. You have one of those on each side. They help with things like pulling the head downward or turning the head. Turning the head would be if you tighten one muscle but not the other one. Let's move on. So the muscular system helps us move, and it's not just move us; it's move blood and food inside of us. All right, so three types: smooth, cardiac, and skeletal. We already said that. The cells, because they're long and thin, they're called muscle fibers. Okay. Smooth muscle. These fibers are kind of like a long tube, but they're pointed at the end. And uninucleated. That's what I'm saying. One nucleus. You might say, what's so special about that? But just wait till we get to the others. Um, they're arranged in parallel lines, forming sheets, but they're not striated. How is that possible? Well, if this one's here and this one's here, and there's not like, see how now my knuckles line up? If you were, say the knuckle is a thicker part of the hand, which it actually is, you would see thicker part and then thinner part, and they'd be lined up. And that might be something that from a distance you could tell, oh, that's where the knuckles are. Not that you could see all the knuckles, but same thing in a muscle. Ah, well, that's the Z line, and that would be what you see in a striated muscle. Uh, Something like the skeletal muscle. Smooth muscles, because the fibers are not all aligned, it's hard to make out where part of this fiber is and part of this fiber is, part of this cell and part of this cell. Anyway, so parallel lines, sheets, not striated. I already said it says the walls of hollow internal organs. Yeah, we're talking mostly the digestive system. We talked about that. And the walls of blood vessels, and that causes constriction of the blood vessel. We've done that. All right, so here we have pictures of smooth muscle. It says the same thing, non-striated, because see, just look at the nuclei. Nothing's lined up. They're all, so there are no distinct lines. There's one nucleus in the cell. It is involuntary. Notice that. So let's go ahead. There are two types of cells that are involuntary. One is smooth muscle and the other is cardiac muscle. Those are involuntary. The skeletal muscle is voluntary, meaning that when I moved those fingers, I sent signals from my brain to my fingers to make it happen. direct control. All right. If you think about it, you can move it. That's voluntary. And smooth muscle is not like that. You can't say, "Oh, I would like to." No, I'm done with that swallowing. I know the food's partway down, but let's stop. No, when you start swallowing, once you get down about here, smooth muscle takes over. It's going to go to the stomach. All right. Moving on. So smooth muscle contraction is involuntary, like I just said. It's slower to contract than skeletal, but it can do it for a longer time, and it doesn't get as tired. All right. Cardiac muscle, that's the heart wall. It says uninucleated, striated, and tubular, but branched. Got lots of branches going in different directions, and intercalated discs. So it's branched, and the parts of this cell will hook to the next cell over, and that spot where they hook together, that's called an intercalated disc. What it is, they're actually protein tubes that go between the cells. They can pass chemicals from one cell to the other. So what that means is if you start a muscle contraction here, the chemicals causing it are going to go into the next one and start the next one. So a lot of people like to say that with heart muscle, you have an all or nothing contraction, because if one contracts, the rest of it is going to try to contract too, eventually, as it spreads through. Now we have ways of controlling that where we have some channels that send the signal faster than others. I'm talking about things called Purkinje fibers, for instance, and we have places where the signal doesn't propagate well at all. I'm talking about from the atria to the ventricle, except in one spot. But we'll get into that. All right, so cardiac muscle, it's got lots of branches. It's still striated. You can see stripes. Here's an example. Here is a stripe. Now those stripes are where several fibers lined up their intercalated discs, and you can see there's another one there. Okay. They've got a nucleus here. It pumps the blood, and it's involuntary. You've heard that before. All right. Cardiac muscle relaxes completely between contractions, which is not much time. All right. That's why your doctor wants you to have a low pulse rate if you can, not too high, because you need time to relax. Okay, the contraction is rhythmic, and it says happens without nervous stimulation, is involuntary. Okay, is involuntary? Yes, I'm going to go with that. I know some people who are, but I can control my heart, and when I'm about to pull the trigger, my heart skips a beat. I think it's a load of hooey. There's no way that we've found that could be done, but whatever. But occurs without nervous stimulation. It would have been better to say it can occur without nervous stimulation. The fact of the matter is that we actually do control the speed of the heart with signals that are sent via nerves. The vagus nerve, if it stimulates the heart, it will slow the heart down, for example. And that's important. Not to be completely gross, but if you work in a nursing home, you might occasionally have to pick up a old man who was straining on the toilet, and because they were kind of constipated, and they black out and fall and hit their head. What on earth? Well, the vagus nerve, that nerve I was talking about that'll slow the heart, it also goes all the way down to the rectum and anus, and passing a bowel movement out, yeah, that definitely stimulates it, and pushes on the nerves and signals on the nerve up towards the brain, but as they do, up towards the heart, and it will slow the heart. And if you're old and constipated and have trouble passing fecal matter, getting rid of waste, might stimulate it enough for you to black out because you're not getting enough blood to the brain. This happens all the time. So occurs without nervous stimulation. There are actually nervous signals that will slow it down, speed it up, hormonal stuff, obviously. But what they're talking about there is that the heart has a pacemaker. There are cells that are self-excitatory. Actually, just like NASA, if you're doing something like, oh, we're going to send someone to space, we're going to have the computer and the backup computer because problems if the primary computer goes out. Same thing. You've got two pacemakers in the body. You have one that is called the SA node, the sinoatrial node, and another one, a backup one called the AV node, the atrioventricular node. And both of those will send signals through. Both of those, they're sitting, they're resting, but they're not really resting. The ion levels inside them are changing in ways that we're about to talk about. And as the ion levels are changing, it's getting closer, closer, closer to contracting. And when it finally contracts, it also sends that signal because of intercalated discs through the cell next to it and next to it, next to it. And they all will contract eventually. That signal to contract will spread from the pacemaker cells through the other cells, and then the heart will contract. And like I said, we have fast and slow paths to do this. That's just because we want to coordinate it. Like when we get to the ventricles, the lower chambers of the heart, we always want to contract at the apex, the tip of the heart, before we do up there at the AV, at the atrioventricular septa. You wouldn't want to contract there because that would be like grabbing a tube of toothpaste in the middle. You wouldn't get all of the toothpaste out. In the same way, if you contracted up there first, you'd never get the blood down at the base out. So fast channels, Purkinje fibers, to carry the signal down and let us contract at the apex first so we can squeeze the blood out of that ventricle. Anyway, but we'll get to it. The important thing here, we're going to talk about cardiac muscle, and we're just doing muscle, is that we have self-excitatory cells in the pacemakers. All right, so skeletal fibers, they're still tubular. They're striated like the skeletal, we are on skeletal muscle. They're still tubular. They're striated like the cardiac muscle, but they're even more striated. Everything's lined up beautiful. And they are multinucleate. They got more than one nucleus. Skeletal muscles are the ones that are pulling on your skeleton, attached to your skeleton. They tend to be super long, and they're under voluntary control. We can choose to wiggle a finger. We can decide what to do with those muscles and when. All right, you see what I meant when I said, oh, wow, they are lined up really, really beautiful. Look at those nice lines. We'll talk later about the different lines, like the Z line and all that. But yeah, so they're lined up. And the reason they're lined up is because all the cells are lined up. Remember the example I used, all my the knuckles lined up. So here, this dark band, that's probably where you have a bunch of thick filaments. This light band, that's probably where you have a bunch of thin filaments. I shouldn't say that. I should just jump ahead and show you. Now I'm going way too far. Here we go. All right. So the light band right here, you've got the actin filaments and not the myosin. Here you got the myosin and the actin. So you have both. You got more stuff going on there. You got more things in that location, and it looks darker. All right, but we'll talk about that later when we do muscle histology. All right, come on. So humans are vertebrates. We got a backbone. Vertebrate just means you got a backbone. So we have a vertebral column, a skeleton, joints in our appendages. Our skeletal muscles attached to our skeleton. Pretend to be shocked by that. And when they contract, it moves the bone, it pulls on the bones and moves the joints, either flexes or extends them. All good. The only thing I want to add there is that at contraction, the muscle fibers are sliding past each other. I just showed you the actin and the myosin. What's happening is they're sliding past each other. And as they do, the muscle is going to get thicker. See, look at this. I'm sliding my fingers past each other and thicker. Okay, thicker. So, and look at the distance between my thumbs. The distance is shorter, so the muscle gets shorter and thicker. That's literally the way it works. Okay. So the function of skeletal muscles, they provide support. They pull on the bones so that you stay upright. They move the bones and other body structures. When they say other body structures, there's the example of breathing. Breathing's pretty important. That's it's all breathing is based on muscle movement, but for instance, what we call the tidal breathing, the shallow breathing that happens at rest, that's all diaphragm. Diaphragm is like this, it contracts and goes down. So why does it contract? Because it gets shorter. So it's this big dome thing, it contracts and it gets shorter, goes down. Well, it goes down, it's pulling on the lungs and the air's flowing in. All right. But other stuff, where it says other body structures. You could have muscles doing things like moving skin. That's the facial expressions. Where it says another function of the muscle, maintenance of body temperature. Think of shivering to stay warm. Obviously, your muscles are releasing heat at that point. They're breaking down ATP and releasing heat, which travels through the body. This is true at any time, but if you get really cold, you're contracting the muscles faster in an effort to fight the heat loss. That's the shivering. All right. Movement of fluids in the cardiovascular lymphatic system. Cardiovascular, you've got that. The heart, it pumps there. Lymphatic, we've probably talked about this before, but if you have a lymph vessel right here and it's between two muscles, remember this muscle is going to get shorter and thicker, and the muscle over on this side is also going to get shorter and thicker. What that means is the vessel in between those two muscles, when you contract those muscles, that vessel gets squeezed, and when it gets squeezed, and you've got valves to prevent the blood from going down or the lymph from going down, that lymph is going to go uphill. So, all right. The muscles can protect the internal organs and stabilize the joints. So they can pad the bones. The muscle wall keeps you from having your intestines like out there and exposed. Gives you more than just a layer of skin covering and protecting them. And tendons hold the bones together at joints. That's true, especially because the tendons help to make they're like the capsule, joint capsule. But I want to remind you that if you hear tendon, the T should remind you that that thing ties muscle to bone. If you're L and ligament, that's because you're linking bones together. So that's how you remember that tendons are attaching the muscle to the bone most of the time. Just use the mnemonic: tendons tie muscles to bones, ligaments link bones together. All right, so now we're going to talk about parts of a muscle. Let's jump up to the pretty picture if they have it. How can you not have it? Okay. Yeah, so if we look right here at this small picture, okay, a myofibril, it's a muscle fiber. They're in little bundles. And if we look at this zoomed up, each fiber, each bundle is wrapped in a sarcolemma. It's a connective tissue membrane. And those are in bigger groups that have a membrane and bigger groups that have a membrane. That's what we're about to talk about, is those membranes. So moving forward, there we go. All right, so a fascicle is a bundle of muscle fibers. And so it's made out of connective tissue. Each fiber has connective tissue, and each fascicle, each collection of fibers, has connective tissue. So there's connective tissue around one, and then you have a whole cluster of them, and there's connective tissue around those. And the fascia that covers the muscles extends, like you have this fascicle, and you have the connective tissue around it that continues off and becomes part of the tendon. And the next one, the next one, the next one. The tendon is not just here is a muscle and here is a tendon and here is a bone. It actually is all of the connective tissue within that muscle around each fascicle coming together. The fascicle narrows, you run out of muscle, but the connective tissue continues all the way down to the bone attached to the periosteum. Okay. You have little fluid-filled sacs called bursa that you can find between tendons and bones. It says here bursa act as cushions and lubrication. Totally true. What I always compare them to is the rope keepers that we used to use when we were rock climbing back when I was younger. I lived in Colorado, and if you were running a rope over a cliff, you didn't want it rubbing back and forth on the rock. If you were moving back and forth, that would be bad for the rope, and you don't want to fall and die. So you had protective things for the rope that were called rope keepers. Well, bursa are like that too. So imagine you're running the rope over a cliff, but before you do, you pick it up and you put a little cushion right there. And so the rope is rubbing on the cushion. That's what the bursa is the cushion. All right, so moving on. Hold on a sec. Okay, let's move on. So the two ends of a muscle are called the origin and the insertion. All right. The origin, it says, oh, attached to the stationary bone. That doesn't mean that bone can't move. It means it's not the bone that's moving right now. Okay. So the attached to the origin, and the insertion is on the bone that moves. When the muscle contracts, when the muscle gets shorter and thicker, what happens is it's pulling on one side of a joint, pulling on a bone that's not going to move, and the other side it pulls on. Let me move this out so you can see better. All right. So here we have, gosh, I really should have brought, we have a joint. Okay. Actually, I can use my hand. That'll work. We have a joint, and on this joint, we have a bone here, we have a bone here on this joint, and we're going to make it an extensor muscle. So it'll attach up here and it'll attach down here. When that muscle gets shorter and thicker, as it gets shorter and thicker, the tendon runs over the joint and it pulls. Okay, so shorter, thicker muscle, tendon, as this gets shorter, it pulls that way and your joint moved. Great. All right, so no big deal. That's not surprising. What I want you to know is that this one, this end of that muscle that's attached, we call the origin, and this end we call the insertion. The origin is on the bone that's not moving, and the insertion is on the one that will be moving as the muscle gets shorter and thicker. Great. So that's fine. The thing that may be unclear to you is that this bone could also be moved by some other muscle. So you could have the insertion on this bone for a different muscle and the origin up there. All right. A good example, easy example. Here we have my elbow, okay? And here we have my wrist, okay? I can move the wrist, and those muscles are attached on here. Okay, get shorter, thicker, get shorter, thicker. Great, and they pull on these bones. But that doesn't mean that I can't also move my elbow. It's just different muscles that are involved. All right, so let's move on. When a muscle contracts, it pulls on tendons at its insertion, the bone moves. That is, when the biceps brachii contracts, it raises the forearm. Here, let's do the thing. When the biceps contracts, it raises the forearm. All right, moving on. So there are names for groups of muscles. I want you to know that muscles work together in groups. The strongest one, the one doing the most of the work for the moving things, moving a joint, that's an agonist. And the one that works with it, like, oh, there is synergy. Lots of people say it's synergy. Well, synergist is the helper muscle that works with it. I'll give you an example. We just talked about what the biceps does. Well, there's an itty bitty little muscle called the brachialis that will also help bend the elbow. But the brachialis is not nearly as strong, generally. I actually know someone who was a professional athlete, and they tore their biceps. Remember, biceps has two heads. The tendon on one of those heads popped off, like on the insertion, it came off. And that what happened? They never got it repaired. I don't know the details why, but they never got it repaired, and the helper muscle, the brachialis, got much bigger and stronger than it would be in a normal person because it was doing the work that part of the biceps normally does. Anyway, so if you have an agonist, then you have the one that works opposite, the antagonist. So your biceps is going to do this. Your triceps get shorter and thicker, and it pulls like that. So biceps, triceps. All right. If both of them contract at once, there would be no movement. I'm going to give you an exception. If both contract at once and can do so with equal strength, there's no movement. All right, so muscles work in pairs. The example they have there, the biceps and the triceps, and it's a good example. Let's go ahead and move on. All right, names of skeletal muscles. They're often used, it says, names often use the following terms to characterize them. You could talk about how big it is. Maximus means, oh, it's the maximum size one. Minimus means, oh, it's the little one. Vastus means, oh, it's vast, it's huge. Longus, oh, it's long. Brevis, it's short. They're straightforward. Oh, it's brief. Okay, or they could be described by shape. The deltoid looks like a Greek letter delta. That's a shoulder muscle. The trapezius is trapezoid shaped. The latissimus is wide. The teres is round. These are just one of those. The more Latin you know, the easier the words are to remember. And if you don't know any Latin and you stay in anatomy, you'll know some Latin when you're done. All right, you could name them where they are. There you go. External or internal, frontalis, pectoralis, gluteus, brachii. The full name of the biceps is the biceps brachii. Sub would be below. Rectus means straight, where it says the rectus abdominis. That's the muscle that everybody's talking about working out to get a six-pack. That's seeing the segments of the rectus abdominis. Orbicularis oculi, this would be the muscle around there. Orbicularis oris would be right here, around the oral cavity, around the mouth. Oblique, that means diagonal, so like your external obliques, your internal obliques. Transverse, yeah, these are straightforward names. So we can name things based on size, based on shape, based on location or direction. We could name them based on where they're attached. I already gave you that example. The sternocleidomastoid is attached to the sternum, the clavicle, and the mastoid process of the skull. Brachioradialis attached to your brachium, to your upper arm, and your radius. That is the smaller of the two bones, the one on the thumb side of the forearm. So right here we'd have our radius, and over here we'd have our ulna. The brachioradialis is attached from up in the brachium down to the radius. All right. Could be based on a number. Here we talk about the quadriceps femoris, 4 origins. Well, actually, the quadriceps femoris is not usually thought of as one muscle, like the rectus abdominis they talk about. The rectus femoris is one of the muscles of the quadriceps. But anyway, moving on. Yeah, you got 4 heads. They're all helping to extend the knee. Moving on. Skeletal muscles could also be named on the action that they take. So an extensor is going to extend the finger. An adductor is going to add things. So we're taking this is over here and adding it to the rest of the hand. That'd be an adductor. An abductor would do the opposite. It would abduct it. It would pull it away. In fact, this muscle is the abductor pollicis longus, the one that pulls the thumb away. Pollicis is a reference to thumb. A flexor would bend a joint. A masseter helps to pull shut the jaws. A levitator would be to lift things up. All right, so here we have a whole bunch of muscles. But let's go over this again. So we could name them based on the size, the shape, the location, the direction, the attachment, the number of parts they have, what action they take. All of those are valid ways to name muscles. It's not consistent. So if you look here on 13.5, that's a lot of muscles. Like, oh my God, but it's a lot less than there actually are. I would say definitely the things on this diagram, figure 13.5, those are all valid things that you should probably know. All right, here is a progress check. Again, you do a check your progress, and if you can't answer it, you roll this video back and you watch that section again. I'm going to pause for a moment. Okay, let's move on. So now we're going to do histology things. We're going to talk about how muscles contract, identifying the structure of a muscle, the sliding filament model. I already told you that you have fibers of actin and myosin. Basically, the heads of the myosin pull on the actin, and that's what causes them to slide past each other. And summarize how activities within the neuromuscular junction control muscle fiber contraction. Basically, it's going to go into more detail. There are more steps than what I'm saying right here. But basically, the nerve signal, the signal from the nerve when it arrives at the neuromuscular junction, is going to cause a special modified sarcoplasmic reticulum that we call, a special modified endoplasmic reticulum that we call the sarcoplasmic reticulum. Sarco just means flesh. So it's like, oh, it's the endoplasmic reticulum in the muscle. And so we must give it a special name. Well, this is endoplasmic reticulum that holds a bunch of calcium. And when the signal comes, that sarcoplasmic reticulum is going to dump calcium. And why do we care? What does it matter? Well, calcium is necessary for the muscles to do their contraction. It uncovers places that those heads can grab on and pull. Well, It binds to another protein, which causes it to move, which causes the uncovering of the places that the heads can bind and pull. You'll see. You'll see. All right. So sarco, remember, it's Latin for flesh. Okay. So a sarcolemma, that's your plasmic membrane. Yeah, of course, they have to rename everything. Your cytoplasm in a muscle, it's got a new name. It's a sarcoplasm. Your sarcoplasmic reticulum, it's the endoplasmic reticulum. And like I just told you, it's a calcium storage site. And T-tubules or transverse tubules, they go into the cell and they get really close to the sarcoplasmic reticulum. What this means is they can carry ions in, carry signals in close to the sarcoplasmic reticulum. All right, so there's a pretty table 13.1. We've already done 1, 2, 3, 4. Yeah, we've done 4 of those things we've gone over. Know the other 3 that are on that table. It's not that bad. All right, here we go. Here's our little model that we were talking about with the myofibril. Okay, and so the cellular parts of a muscle fiber, the sarcolemma has many myofibrils in it. Let's back up to make this make sense. The cytoplasm of the cell has a bunch of muscle fibers of a muscle cell. It's got a bunch of fibers in it. Great. So it has a bunch of myofibrils. These are the parts that contract. It also has glycogen, as in sugar molecules. Remember, a long time ago, we talked about how glycogen is the animal energy storage molecule. It's a bunch of sugars all hooked together. And this is what provides the energy for muscle contraction. The and I'm going to give you an exception. Cardiac muscle. The heart at rest, not when it's working faster than normal or something like that, but the heart at rest runs on fatty acids more than sugar. You have some tissues like the brain that only run on sugar. Anyway, moving on. The sarcoplasm binds myoglobin, or includes myoglobin, which binds oxygen. Let me break this down for you. Myoglobin is like the muscle form of hemoglobin, all right? It's red and it stores oxygen. And how much oxygen it stores controls what you can do. And I'm not saying that just like some random thing. One time when I was in graduate school, I helped dissect a dolphin. And you know, dolphins can hold their breath longer than we can and stay down longer than we can. How? Well, their muscle has so much myoglobin in it that it looks purplish black. I mean, it's not like, oh, it's kind of red. It's more like red food coloring. You know how red food coloring, when it's so concentrated red that it looks blackish? It's like that, okay? All right, so muscle fibers are cylinder shaped, and inside of the muscle fiber, you have a bunch of little myofibrils. These run the entire length of the muscle. And we're talking striated muscle mostly. And each myofibril is made out of myofilaments. So you have a muscle fiber that has myofibrils in it, that has myofilaments in it. And remember, you got connective tissue around these things that turn into, as they continue on, the tendons that do the attaching. So striations are because of the placement of the myofilaments within the myofibrils. And like I told you before, they've got to line up just so if you're going to see stripes from a distance. The thick myofilaments are myosin, and the thin ones are actin, which are the tiny little microfilaments inside normal cells. And you already know the myosin have heads, and they grab onto the actin and pull on it. They're pulling on the actin, and that's what causes the muscle to get shorter and thicker. All right. So myofibrils are divided into sarcomeres. A sarcomere is the distance from one Z line to another Z line. Well, what am I talking about with Z lines? Let's go back. There's probably a picture coming up, but this would be a Z line right here. What it is a place where you've only got actin. There's no myosin there at all, just actin. You're like, well, what does that matter? Well, it matters because if these myosins are pulling on those, and these myosins are pulling on those, the muscle's going to get shorter. You're going to fill in the space. This space right here is going to get a lot narrower as they pull in, as the myosins pull in. All right. Moving on. So sarcomeres extend between two dark vertical lines called Z lines. The I band, which is light color, is only thin. The A band, overlapping thick and thin. All right, do we have a good picture coming up? Yeah, it's a mediocre picture, but we got one. All right. So I'm going to jump up to here. I was about to go back to the other one, but right here we have that Z line where it's got actin. Okay, Z line. Then we have just actin. So the Z line is named Z line because you got a zigzag back and forth there. And then you have this light area because you got less stuff. You got nothing but actin, and that's what we call an I band. Okay. Now, the opposite of that would be an H band, where you have nothing but myosin. Okay. And the myosin you see right here, it's a thicker fiber, and it has lots of these little heads on it. Those heads are what are going to grab onto the actin and pull on it to narrow it, to narrow the muscle, make the muscle shorter. So here we have a relaxed muscle, and you can see big I bands, big H band. When the muscle contracts, the myosin pulls on the actin, and that actin, that Z line, moves in. Okay. And when that happens, the I band gets narrower. That's a fancy way of saying the myosin fibers, as they pull the actin in, they get closer to the Z line, so the I band disappears. All right, let's move back. Okay, so the I band was light colored, only the thin filaments. The A band, overlapping thick and thin. I didn't point that out. I should have. Okay, here we have the A band where we have thick and thin fibers right there. All right. And you might say, but there are no thin right there. There are when you're contracted. So at least some of the time their definition works perfectly. All right, let's move on. Centered within the A band is the vertical H band, which is only thick myofilaments at rest. All right, so the thick filaments are myosin. Each myosin molecule, like a golf club, and the end that sticks out of it is the head. Cross bridges, I've always heard people use that term for the type of bond they're going to make. But anyway, you have this head that sticks out. Here, imagine we have a myosin molecule, and here's the head on the end of it. Well, you're going to have lots of those, but those heads that are hanging down, they're what will grab onto the actin and pull it. All right. So thin filaments are, it says here, actin with tropomyosin and troponin. This is important. This is where we're about to get into all of that calcium. Assuming your book goes into that much detail. Yeah, I'm not seeing it. Yeah, they might. Yeah, they are. Okay, so right here. I'll back up, but to where we were supposed to. But here we have that pair of twisted fibers. Those are the actin fibers. So 2 actin filaments attached, but you have troponins, little binding sites attached to tropomyosins. Okay, I shouldn't say little binding sites. You have little troponin molecules, and they are attached to tropomyosins. But here's the deal: when calcium is released from the sarcoplasmic reticulum, and it binds to the troponins, the troponins move a little bit, and because they're attached to tropomyosins, the tropomyosins move a little bit. And now you see these dots that when we had no calcium, they were covered. I mean, you can see that they're behind the tropomyosin. When calcium binds, everything moves a little out of the way, and it exposes binding sites that the heads are going to grab onto and pull. So you need calcium in order for the muscles to work. All right, let's move on. We'll move back so we get to the... So thin filaments are made out of 2 twisted pair of actin. And on top of that, you have the troponin and the tropomyosin. Let's look at it again. And what you see right here, the troponin and the tropomyosin. You might say, how do you remember which one? Well, tropomyosin is the longer one. It's the one that's like a big rope going down the whole thing. So the longer word is the longer protein. It's the tropomyosin. The shorter one that's just right here is the shorter word. It's the troponin. So there's a mnemonic to help you remember it. All right, moving on. We've talked about the 3 parts of a thin filament, and you already know, I sort of spoiled it, but you already know that what happens is you uncover binding sites. When the troponin moves, it pulls on the tropomyosin and uncovers binding sites that the actin is going to use. All right, so the sliding myofilament model. Muscle fibers slide around, move past each other. When we contract, the sarcomeres get shorter. Because these things go the whole length of the whole muscle gets shorter. All right, so the thin filaments are going to slide past the thick filaments, keep the thick filaments in place. The I band will get shorter as the Z lines are coming in. And the H band in the middle, where you had just myosin, no actin, that disappears as the actins come to the middle, as the thin filaments come to the middle. Now, you already know this. You shouldn't be surprised. Muscles need energy. And like most of the body, most of the cells, ATP is the energy source we like to use. So muscle filaments break down actin, and the cross bridges at the head of the myosin, pull the actin filament towards the center of the sarcomere. You've heard me say it many times. The myosin is pulling on the actin and bringing that Z line in, pulling on the actin, bringing that Z line in. As we bring 2 Z lines together, the whole muscle is getting shorter and thicker. All right. So there's this picture that we keep coming back to. We're great. So now let's talk about the contraction. You have a motor neuron. This is a neuron, comes from the nervous system, that tells the muscle, oh, you need to contract now. A nerve is a group of neurons. That's great. Whatever. The axon is like a long electrical cord. You send an electrical signal down it, electrochemical signal actually, down it. And you have a little bit of branching on that axon, so it can talk to several different muscle fibers. You don't have one axon goes to one muscle fiber. You have one axon comes in and it's going to get that muscle fiber and that one, so they're all contracting or all get a signal to contract. All right, so neuromuscular junctions. Here we have muscle fiber, okay? And you can see the axon is branched, and we have signal coming to this fiber, and signal going to that fiber, and signal going to a fiber down there. The end, the terminal, is a big wide area, kind of a discus attached. Okay, so an axon, part of the nerve that carries a signal, goes to muscle fibers, skeletal muscle fibers, and it ends in a terminal that's touching or very, 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 very close to that muscle fiber. All right. Where an axon terminal, the end of the axon gets really close to the sarcolemma. Remember, the sarcolemma is the plasma membrane of a muscle cell. All right. You have a little space between the nerve and the muscle. We call that the synaptic cleft. That's a little space that is going to the end of the axon terminal. So we have the nerve signal coming down. We have that terminal at the end. This end, this disc that's super close to the muscle, it's going to have in it a bunch of little vesicles full of a neurotransmitter called acetylcholine. So when a signal comes down the nerve axon to a terminal, it's going to cause little sacs, little vesicles to fuse with the plasma membrane and dump acetylcholine into the space, the cleft, which is the space between the nerve and the skeletal muscle. So here you see it in action. We have a synaptic bouton in the terminal. We have a synaptic vesicle full of acetylcholine. It comes up, it fuses with the plasma membrane, it dumps out the acetylcholine. The acetylcholine goes across the synaptic cleft, and it's trying to bind to receptor molecules here. All right, so the acetylcholine goes across the synaptic cleft, binds to receptors, and that causes an electrical signal to go across the cell plasma membrane, down the T-tubules to get close to, very close to, the sarcoplasmic reticulum, which you remember is just muscle cell endoplasmic reticulum that is being used to store calcium. What that electrical signal does when it gets close is it causes the signal, the electrical signal causes the sarcoplasmic reticulum to dump calcium. All right. We've already talked about the other fibers. You know that you have tropomyosin, the long one wrapped around the actin. You know that you have little troponins on it. When calcium is released from the sarcoplasmic reticulum, it's going to bind to the troponin. That's going to make the tropomyosin move slightly. And when it moves, it uncovers heads on the actin uncovers binding sites on the actin that the myosin heads can grab onto and pull. All right, so the sliding myofilament theory, this is how we think this works. The myosin heads have ATP binding sites. ATP binds there and it's broken into ADP and phosphate. That's pretty straightforward. That's how we get energy, is releasing a phosphate that doesn't want to be there. The myosin heads are going to grab to the actin at those binding sites. They form cross bridges, just links between the myosin heads and the actin fiber they're going to pull on. You then let go of the ADP and the phosphate, and that causes the heads to bend. Okay, that's what pulls and causes the fibers to slide past each other, causes the muscle to get shorter and thicker. Okay, so that's powerful. And here we go. And then if you break fresh ATP, things will go back. All right. The binding of the ATP to the myosin head breaks the cross bridges. Myosin will let go of the actin. The cycle begins again. It reattaches further down. This will happen again and again as you shorten the sarcomere, shorten the muscle. If you can't relax the muscle because you run out of ATP, then you have permanent stiffness of the muscle, permanent contraction, shortening. That's the rigor mortis they're talking about, which is common when people die and they run out of ATP. The muscles stiffen up. All right, let's move on. There is a memory check here. I will pause for a moment and you can see if you can answer these questions. If you can, that's great. If you can't, rewind, watch it again, and then we'll move on. Okay, let's keep going. So now we are going to do more discussion of muscle contraction. This is talking more, instead of the histology and the sliding myofilaments, this is talking more about the whole muscle and the activity of the whole muscle. We'll talk about muscle twitch and probably talk about how there's slow twitch and fast twitch muscles. Summation, that's when you get multiple signals and how the muscle responds as a whole to multiple contraction signals. Tetanus or tetanic response, that's when there's a constant pull with the muscle. This happens because you never contract the whole thing at once. You have some fibers that are relaxing while others are contracting, but you always have something going on. So there's always tension. That's tetanus condition, or tetanic condition. And we'll talk about how the muscles make ATP that they're going to use. And oh yeah, they're down at the bottom. Talk about the difference in fast twitch and slow twitch muscles. Moving on. All right. So muscles have motor units. Okay, so a motor unit is a nerve fiber and all the muscle fibers that it innervates. That doesn't always necessarily mean it's the entire muscle. The number of muscle fibers in a motor unit can vary. Some you have only a few fibers that are being stimulated. Some you have a huge number that are being stimulated. The muscles that are used for very fine control, small movements like signing your signature, those tend to be fewer motor unit, fewer muscle fibers in a motor unit. All right, the ones that have lots are strong, big movements, strong muscles. All right, so a muscle twitch is the contraction of a muscle fiber. It's very, very brief. And let's talk about what it takes to do it. You have a latent period. That's the time it takes between the stimulation coming from the nerve and the contraction starting. What's going on there is that acetylcholine is going across the synapse. An electrical signal is coming down the sarcolemma, the plasma membrane, and down the T-tubules. Remember, the T-tubules stick down in, so you have a piece of plasma membrane and these transverse tubules that go down. What they're doing is carrying that electrical signal down super close to the sarcoplasmic reticulum. Fancy name for the endoplasmic reticulum, specialized to hold calcium. All right, so the second stage is contraction. The calcium is going to come out of the sarcoplasmic reticulum and cross bridges form. Let's be more specific. The calcium will come out. The calcium will bind to the troponin, which will move the tropomyosin out of the way, exposing the binding sites for the heads of the myosin, and the cross bridges will form. And then we have a relaxation period where those cross bridges are broken and the calcium is pumped back into the sarcoplasmic reticulum. And the strength, the force will diminish as the muscle's relaxing, returning to its normal length. Remember, when the muscle contracts, it gets shorter and thicker because the fibers are sliding past each other. All right. So let's move on. Yeah. Here they are showing in a muscle twitch how you have the latent period where the signal is spreading, the contraction period as the calcium is being used, and the relaxation period where you put everything back. Summation is when you have more muscle contraction. You might say, how? It was, you said earlier right here, they either all contract or they don't, all the fibers in a motor unit. Yeah, but muscles have more than one motor unit. So, maximal sustained contraction is tetanus, and that'll continue until the muscle tires out, runs out of energy, or so that can continue as the muscle tires, runs out of energy, and then can't do anything, even though you're stimulating it. Where they say, oh, not the same as the infection called tetanus. That's kind of accurate, but kind of misleading because the infection causes tetanic contraction. So, yeah, they're right. The infection is different, but the chemicals released in the infection cause this type of contraction. Anyway, let's move on. So here you see you're getting a stimulus, you're getting the force increases as we go. So stimulus to contract, we get some force, we're starting to relax and go back, but before we can, we get another stimulus. So we contract more, it's a little stronger. And then we get another stimulus to contract. And it goes even higher, more force, and more, until the muscle is as contracted as it can be for as long as it can go. And then it tires out, and it doesn't matter how many more stimuli you're sending. Here's a stimulus. You can see you're sending lots of stimuli. Well, you're still doing it here after the muscle is in fatigue. It's just being ignored because the muscle is tired out. Okay, recruitment. What recruitment is, you saw how the contractions here, we had a stimulus and it's starting to relax. And before it could relax, we had another stimulus and we got another contraction, but it was stronger than the first one. If you think about it, it makes sense. Say you have a muscle that's this long and it contracts, so it gets shorter and thicker. Well, when you relax, it's starting to go back. But what if what if first contraction, you're this wide and you contract to this wide? Now you're relaxing, you're going back, but there's another stimulus, so you contract even more. Because you were already contracted, you're going to go further. So there'll be more force generated. And that's what they're talking about with recruitment. It's because you're adding motor units. So as the intensity of the nerve stimulation increases, more motor units in the muscle are activated. That's the recruitment. You're adding more motor units, which means more muscle fibers are contracting. The maximum contraction requires all the motor units, and this usually doesn't happen because they would get tired all at the same time. And we try to avoid that. It says instead, some motor units contract maximally while others rest. That is the tetanus thing that they're talking about. You will have you will constantly generate some force because some muscle fibers are in motor units that are contracting while others are resting, and then they'll trade places. So muscle tone is how firm your muscle is, and it depends on muscle contraction. You always have some contraction happening, but usually it's really slight, not enough to cause muscle movement, but it can cause tone, muscle tone, tension, a little bit of tension. We're now going to talk about bioethics, performance enhancing steroids. Steroids that make your muscle tissue bigger by promoting protein. They're not saying it here, but I'll go ahead and say it. Anabolic, that's just a general term that means we're adding things together. In this case, adding amino acids together to make a protein. That could let us grow more motor units. That is an anabolic steroid, okay? We're talking about steroid hormones that occur naturally, and it says most closely related to male sex hormones. They're talking about testosterone and the other androgens. All right, so this can increase the size of muscles. It can give them more endurance. You can store more glycogen for energy in them. But high blood pressure, acne, and cancer. And in women, if you're taking a bunch of those male hormones, your voice gets deeper, you develop more body hair, you have changes in your genitalia, your menstrual cycle changes. In children, it can cause early puberty. It's not good stuff to mess with. Okay. And you might have heard people talk about roid rage. Roid rage is roid short for steroid. It's violent mood swings when you're on steroids. And a lot of times these things can come up when people are withdrawing from the use of steroids. Also, if you're always taking drugs for steroids for performance, you might feel like it's cool to take other drugs. It can be a gateway drug causing a serious drug problem. Actually, that combined with painkillers when athletes overexert themselves, the combination of the two. But moving on. All right, so there are 4 sources of energy for a muscle. And the glycogen and the triglycerides, the fats, we can store in the muscle. The glucose and the fatty acids we get out of the blood. Now, you probably already know that glycogen is made out of glucose and that triglycerides are made out of 3 fatty acids hooked to a glycerol. So, yeah, they're listing 4 things, but the way it works is these things get taken in. If you can use them directly right now, great. And if you can't, you can store them up as glycogen or triglycerides to use them later. Which one you're using depends on how intense, how strong the exercise is, and how long it goes on. If you exercise for a long amount of time, you're going to use up any energy stored in the muscle, and you're going to have to pull the energy that's available in the blood. All right. So pretty picture, and you can see that if you're exercising a small amount of time, you're mostly going to be using glycogen out of the muscle. That's why people carb load, is to build glycogen in the muscle. If you've been exercising 4 hours nonstop, it's mostly going to be fatty acids that you're pulling out of the blood. Anyway, let's move on. So muscles can store a very small amount of ATP. I say very small because ATP is an unstable molecule. It probably is going to last, if I remember correctly, about 10 minutes after you stop the production of ATP. Once you use up ATP, there are 3 ways that you could make more. One is creatine phosphate. Another one is fermentation. This is what you do if you're not able to take in enough oxygen and you have to run some of the cycles without oxygen. If you do that, you're going to build up lactic acid. Yeast would build up alcohol. Woohoo! Cellular respiration is the third one. This is the method that would be the best. If we can take in enough oxygen to supply the mitochondria, That makes the breakdown of sugar a lot more efficient. We get a lot more energy out of it if we can run through the mitochondria. But like I said, cellular respiration, going through all three steps, glycolysis, Krebs, electron transport chain, those last two, Krebs cycle, also called citric acid cycle, electron transport chain, also called oxidative phosphorylation, those two things both need oxygen and they happen in mitochondria. So let's move on. All right, so the creatine phosphate pathway can make some ATP. That can happen without using oxygen. Sorry, wrong way. Let's see. Okay. Yeah. So creatine phosphate pathway, that'll work. The fermentation pathway, that'll work, but you're going to build up lactic acid and it's going to hurt. Aerobic respiration, which happens in the mitochondria, that generates a lot more ATP. I wish this picture had like ATP, ATP, and then boom, huge ATP for the aerobic respiration because it's a lot more efficient. It creates a lot more ATP, but it needs oxygen. All right, so creatine phosphate is the simplest and fastest way for a muscle to get ATP, and it's one reaction. You break down creatine phosphate into creatine, and in the process, you turn ADP into ATP. Ta-da. All right, creatine phosphate is formed when a muscle is resting and you store a little bit of it. It's what you're going to use at the beginning of exercise. It says that is the energy to complete a play in a football game comes from, they should say from the first play of the game, comes from creatine phosphate. If you're doing something that lasts longer than 5 seconds, and you're not getting enough oxygen, you're going to do fermentation. That is the lactic acids method. So the anaerobic process of glycolysis and fermentation produces 2 ATP from the breakdown of glucose to lactate. Let me break that down for you, actually. Glucose is a 6-carbon sugar, and glycolysis breaks the glucose into 2 pyruvates. If we have enough oxygen around to be able to further break this down, we'll turn it into acetyl-CoA, and then we'll go through the Krebs cycle and oxidative phosphorylation, all that happy stuff. If we don't have the oxygen, then we are going to turn the pyruvate into lactate. And the way that we do that is to take the pyruvate and put, when we made NADPH before we made the ATP, we were all happy about doing that. But we'll go stick those hydrogens and electrons on the pyruvates and turn them into lactates so we can get the NAD plus back. You got to have that NAD plus or you'll never get to the making of the ATP. Anyway, so you use hormones that break down the glycogen into glucose. Then that glucose is broken into the 2 pyruvates, and it says fermentation is fast, but you build up lactic acid, and that causes muscle aches and fatigue. We know that. And the reason why you're still exercising after you're done, or so the reason you still breathe hard after you're done with the exercise. Say you run wind sprints, and when you're done, you're still breathing hard. It's because you're in oxygen debt. Heavy breathing that's after the exercise, it's needed to get the oxygen to turn the lactate back into pyruvate and then into acetyl-CoA and run it through the Krebs cycle. So that's what you need to, you need the extra oxygen after the exercise to get back to where you should be, and then you can do things the right way, the normal way, and get all the energy out of that sugar. If you do a lot of exercise, you're probably going to have a lot more mitochondria than other people in your muscles. Now, cellular respiration, this is going all the way through glycolysis, all the way through the Krebs cycle, all the way through oxidative phosphorylation, the electron transport chain. It's the slowest of all those because you're going through 3 whole processes. But it says the most efficient. I'll translate. Instead of getting like 2 ATP out of a sugar molecule, you're going to get like 36, 38. Huge difference. Okay. This happens in mitochondria. It needs oxygen. Myoglobin stored in the muscle will give its oxygen to the mitochondria, and the mitochondria are using pyruvate from the glucose. But where does the glucose come from? The glycogen, and that could be glycogen stored in the muscle that's broken down for glucose, or could be pulling the glucose directly out of the blood. You could also use fatty acids. That's a more elaborate pathway. But what you're basically going to be doing is breaking the fatty acids down and using them as intermediates partway through the pathway that would be used for sugar. All right, let's move on. So you have fast twitch and slow twitch muscle fibers. You've probably heard this. Fast twitch fibers use creatine and fermentation. They don't need oxygen. Slow twitch fibers prefer to do the whole thing, the glycolysis, the Krebs, the electron transport chain. And that means they are aerobic, they need oxygen. All right, so fast twitch muscles, weightlifting would be an example. It's anaerobic. Slow twitch muscles, long distance running would be an example. You're needing the oxygen. All right. The fast twitch ones they're showing as light color, and the slow twitch ones is dark color. Every muscle can have some of each, but different muscles and different muscles in different people can have different mixtures, different amounts. All right, so the fast twitch fibers are for strength, and we already said that way back when we first started talking about motor units. Some motor units, ones for fine control, have only a few fibers. Motor units for strength have many fibers. This is where this comes into play. So fast twitch fibers are for strength, and fast twitch fibers, their motor units have lots of fibers in them. Explosion of energy, this is what you see in sprinting or weightlifting. Fast twitch fibers are light colored because they don't have as many mitochondria because they're not using the oxygen. They don't have much myoglobin because they're not delivering oxygen. They have fewer blood vessels because they're not depending on oxygen. They develop tension faster than slow twitch muscles, and the maximum tension is greater. So these things contract. They contract fast and they contract hard. Because they depend on anaerobic means, they're going to tire quickly, and you're going to build up painful lactic acid when it happens. Now, slow twitch fibers, they're in motor units that are smaller, fewer muscle fibers, more stamina. You can keep going longer because you're depending on oxygen. And that means that you're getting more ATP out of every glucose that you break down. Most of their energy comes aerobically using oxygen, and that means the 2 processes in the mitochondria. So we're talking about the Krebs cycle, also called citric acid cycle, and the electron transport chain. They only tire out when they run out of fuel. and they have lots of mitochondria, lots of myoglobin, so they're using lots of oxygen with the mitochondria. They're delivering lots of oxygen with the myoglobin. Let's move on. All right, because they need lots of oxygen, they've got dense capillary beds delivering lots of blood so they can get the oxygen. They will contract a little more slowly, but they're much more resistant to tiring out, to fatigue. And they do have glycogen and fat stored inside. So the mitochondria have a supply of fuel to work with. And if they're getting oxygen, they can make lots of ATP that way, burning the fuel they have stored inside. All right, here's a check your progress. I'm going to pause for a second and you look at these questions, see if you can do them. If not, rewind, watch again. And I will have soda. Okay, so let's move on. We're now going to talk about problems with muscles, muscular disorders. We'll talk about sprains, strains, things like that. Fibromyalgia, muscular dystrophy. You'll see. Let's go through it. So spasms are muscle contractions that you are not controlling, and they hurt. Convulsion, you could have them in smooth muscles, you could have them in skeletal muscles, which are supposed to be under voluntary control, but not always. So convulsion would be a fancy word for muscle spasms in skeletal muscles. Cramps would be strong, painful muscle spasms, and those usually come, it says, due to strenuous activity. All right, we'll just run with that. There are cramps you can get for other reasons too, though. Facial tics would be you have a muscle spasming in the face, and so you have your little twitch, weird motions in your face. Those are skeletal muscles, so you can try to control them, but it's difficult. It's a pain in the butt. Now, a strain is if you stretch or tear the muscle a little bit. A sprain, with a P, you twist the joint hard enough that you got swelling and injury to muscles, ligaments, tendons, blood vessels, nerves. I know that's straight off the slide, but let's go over this. If you twist the joint, remember the joint capsule, if it's a synovial joint, is made out of the ends of the tendons coming together. So that's why you're going to mess up the tendons. That twisting is probably going to pull on the bones. And remember, you've got ligaments that are linking bones together. You have to deliver blood vessels and nerves to the bones. If you're twisting and pulling them apart, you are pulling on that stuff. Anyway, tendinitis. Itis is inflammation. Anytime you see the word itis, it means inflammation. So tendinitis is inflammation of a tendon, and it could irritate the little fluid-filled sac, the bursa, that the tendon is running over. Remember I said those are like rope keepers to protect the rope climber's rope. So the tendons, the idea is the tendon is rubbing on the bursa instead of rubbing on the bone and getting worn down. But if you have the tendon inflamed, it may be bigger and rubbing more on the bursa, and then it can cause inflammation of the bursa, bursitis. All right, so myalgia, just a fancy name for your muscles hurting, and often it happens because you're using that muscle more than you should, or certainly more than it's used to. And that's why sometimes you do something and your coach will say, oh, back off for a little while, let it recover before you do that intensity again with that muscle group. That's also why people work out a lot. It's like, oh, today is leg day. It's not every day is leg day, because you got to have a recovery. Myositis, remember itis, inflammation, myo, muscle. This is inflamed muscles. This is a bigger deal. This is not good. This could be a viral infection. This could be a problem with your immune system. Fibromyalgia is a long-term thing. Your muscles are painful. You can feel stiff. And we don't always know what causes it. And my guess is that it's a term for a symptom and that there are probably multiple different causes for it. There are a lot of things like that. When we talk about neurology and nerves, they'll probably talk about epilepsy. Well, epilepsy is, well, they have seizures, but there are different causes and they're treated in different ways. We're not always sure what's going on. Anyway, so let's move on. Muscular dystrophy. Dystrophy is things are changing. They're abnormal. So it's a group of disorders where the muscles are breaking down and getting weak. Duchenne's muscular dystrophy. It's a genetic disorder, and you're having trouble making dystrophin. Calcium will go into the cell. Now remember, normally we want the calcium, we store it in the sarcoplasmic reticulum, and we use it to cause contractions. In this case, the calcium goes into the cell, it activates an enzyme that will dissolve the muscle fibers. So the muscle fibers are wearing down due to this genetic disorder. They're being broken down. Okay, myasthenia gravis. It's an autoimmune disorder. The muscles in the face, neck, and the arms and legs, they're getting weaker because the immune system is making antibodies that destroy the receptors on the muscle for neurotransmitters. So the nerves are still sending the signal, but the muscles are worse at recognizing the signal and responding to it. So how do they treat it? They give you drugs that will slow down or stop the enzyme, inhibit the enzyme that digests the neurotransmitter. And that means that the acetylcholine will build up in the synapse and make it easier for to get enough of it to cause a contraction. Sarcoma is a word for cancer. Okay, cancer of the muscle here. Muscle tissue or connective tissue of muscle. And it could happen in bone. That would be an osteosarcoma. Adipose tissue would probably be a fibrosarcoma, I'm guessing. And obviously cartilage chondrosarcoma. But let's go through these. All right. Actually, that's the end. That's all the deeper they wanted to go into that. So check your progress. Make sure you know the difference in a strain and a sprain. Be able to compare and contrast the causes of myalgia and myasthenia. Know what some of the symptoms are of muscular dystrophy. I'm going to give you a second. If you can't get this, roll the video back, watch it again. And let's move on. Homeostasis is maintaining internal body, maintaining the inside of your body under constant internal conditions. And the role of the muscles in the body is mostly for temperature. All right, so muscles and skeleton work together to allow you to move the joints, to move the whole organism. Skeletal muscles contract, pull on bones. You saw me doing that many times when I was like, oh, and this muscle right here, this triceps, it'll contract and it'll pull on the bones of the forearm, and that will extend the elbow joint. You saw that kind of thing. That's what they're talking about. And the ability to move our joints and move around in the environment lets us respond to changes in the environment. Contraction of skeletal muscles is also necessary for chewing, moving your mouth, swallowing, moving food to the larynx, Once you get food, why did I say larynx? Pharynx. Moving food to the oropharynx and the laryngopharynx. Once your food gets down into the esophagus, then it's no longer skeletal muscle. That's smooth muscle doing the peristalsis to carry it down. That's why we can't change our mind halfway through a swallow. If we get something partway swallowed, it's going to finish the swallowing. But anyway, muscular and skeletal systems. Rhythmic, smooth muscle contractions move food through the digestive tract. That's what I was just drawing. The peristalsis in the muscle wall as the circular contractions occur. Also, the cardiac muscle, when it beats, you move heart. When the muscle of the heart beats, you move blood around through the body. And it says contractions of skeletal muscles push blood towards the heart. Again, we've talked about that. Think the veins and the lymphatic system, where you have a lymph vessel or a vein right here, and you have a muscle and you have a muscle, and each one of those muscles contracts and gets shorter and thicker. And when they do, they squeeze the vein or the lymph vessel, and that makes the fluid want to go both directions, but it can't go down because you've got a valve. So the fluid can only go uphill. This makes the blood go up against gravity towards the heart. This makes the lymph go up against gravity towards the the ducts, like the thoracic duct, where you're going to dump it back into the circulatory system. And also it says squeeze interstitial fluid into lymphatic capillaries. The wall of a lymph capillary is almost, it's like made out of cells that are only attached on one side so they can all move. And what that means is that the entire wall is like one-way valves. So fluid can go through and now it's trapped. It could go through from this side over here in. It can flow into the vessel, and then when pressure increases, it's trapped. So that's what they're talking about with the muscle squeezes the interstitial fluid and it goes into the lymph capillary and then it's trapped. All right. Here is a figure 1313, and it's talking about how all these systems work together. But the muscular system is the one we're focusing on. All right, so let me move that so I can read it. Both systems, muscle and bone, protect the body. The brain, the heart, the lungs, spinal cord, kidneys, liver, they're in the skeleton, okay? Skeletal muscles pad and protect the bone. The muscles of the abdominal wall, like I'm talking below the, when we're up here, we got the ribs to protect. But below the ribs, all you have is that the muscle wall is the wall of the body is made out of muscle and it's thick, and that protects things like the intestines that are inside. All right, and moving on. Muscles help regulate body temperature a couple different ways. When we're cold, the smooth muscle contracts and it keeps the blood from going up to the surface, to the skin. It doesn't get cooled off by the cold water or the cold air that we're in. It keeps the body core warm. Okay. In extreme conditions, it will do that so well that it'll cut off blood flow to tissues and you'll lose a finger or a toe. We call that frostbite, but it'll keep you alive by keeping that warmth inside for your nervous system, and that's good. One of the things it'll do before that happens is shivering. Your skeletal muscle, if you get too cold, it's going to start quivering. Da, 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 da. This is going to use energy, use glucose, but as you use up energy, you are going to make ATP and you're going to make heat. Think of the quivering muscles when you're shivering as being kind of like rubbing two things together to build up heat. It's the same goal. It's to build up heat because you're moving the muscle fibers, you're building up heat. If you are really cold, you also can get goosebumps. Now, that doesn't do squat for us because we don't have enough hair, but it is a leftover of the erector pili muscles pulling on the hair follicles to stand the hair up. Why would that be useful? Well, if you're a dog and you've got a lot of fur, all that fur stands up and now you've got hair trapped between the fur and the skin. Well, the body heat can warm up that air that is trapped and doesn't easily escape and move away. You or I, if we have exposed skin and we're outdoors, our body tries to heat up that air, and then that air moves away and we've lost the body heat forever. And that's why we get cold. If a dog is out in the cold, or any furry mammal or fluffy mammal, I mean, it doesn't have to be a bird will do it too. They fluff up their feathers. The idea is raise them up off the skin a little bit, trap air underneath. You heat that up the same way, but it doesn't escape and go away because it's trapped underneath the fur or the feathers. It's the same idea that humans use if we're in cold water when we wear a wetsuit. Our body warms up the water, and then instead of letting the water leave and go away, it's trapped around us in the neoprene. So anyway, goosebumps are an attempt to raise the fur, but we don't have enough fur for it to be useful. So, nice try. Okay, there is a check our progress. I'm going to add something here. Oh, and that is the very end. So we're done. But I'm going to add something here because all of this maintain body temperature stuff, it was talking about what we do in the cold. Let's talk about what you do in the heat because that's still muscles controlling temperature. If it's too hot, if you're too hot, you are going to use smooth muscles. I'm talking about the precapillary sphincters to open and send blood through the capillaries in the skin. That has them at the surface and it will help radiate heat away from the body. Better, you work with the skin sweats, the sweat evaporates, you have evaporative cooling that is helping to reduce the temperature of the blood in those capillaries, and then you deliver that nice cool blood back to the body core. You've reduced the temperature by opening the smooth muscles, the precapillary sphincters, which are made out of smooth muscle. Anyway, okay, so we've got that, and now we're done, and I will meet with you guys again for the next chapter. Anyway, talk to you later.